We'll ask us a question of Gabor, I think, first. I think it actually works. So go ahead, Dan.
Okay, it is my pleasure to introduce Gabor Foldvari. He's a research group leader at the Institute of Evolution Center for Ecological Research in Budapest. His research focuses on the ecology of emerging infectious diseases. He obtained his PhD and habilitated in veterinary parasitology at the University of Veterinary Medicine in Budapest, but he has teaching experience in zoology, parasitology, and parasite ecology in teaching in English, German, and Hungarian at various universities throughout Europe. He's been a guest lecturer in the veterinary universities of Madrid, Brno in the Czech Republic, and Vienna, and has had research fellowships at the Complutense University of Madrid, the Konrad Lorenz Institute in Vienna, and the Natural History Museum in London. He works with a broad range of international collaborators in parasitology, disease ecology, and especially in the ecoepidemiology of ticks and tick-borne pathogens and the effect of climate change and biodiversity loss on emerging infectious diseases. He leads the Emerging Pathogen Ecology Research Group at the Institute of Evolution Center for Ecological Research in Budapest. He also investigates preventive measures and application of the DAMA protocol for ticks and tick-borne pathogens as leader of the Ecoepidemiology Division of the National Laboratory for Health Security of Hungary, and he chairs the EU-funded cost action program called Pragmatic. Quite an accomplished young man, and it is my pleasure to call him a friend as well as colleague. Take it away, Gabor.
Thank you very much, Dan, for the nice introduction. And thank you, Scott, also for the opportunity to talk to you at the mentor seminars today. I will start with sharing my screen and hope it will work.
Yeah.
Okay, so you can see the screen now, I suppose.
Looks great.
Okay. So my title is Citizen Science and the DAMA Protocol Can Help Us Cope with Emerging Ticks and Tick-borne Pathogens. And first of all, I would like to thank for my collaborators and colleagues, my grad student, Eva Szabó, who is also with us today, Gabor Kemenesi, a virologist, friend and colleague, also in Hungary, Stottmari, who was inspiring me to continue with submitting all the many proposals until I win, and I win again. And of course, then Daniel Brooks, who was supportive after the time we first met, actually many years ago, and whom I can thank all this conceptual framework that I am right now working with. And I have the luck and the honor to lead two different big projects currently. One is called National Laboratory for Health Security, and within this big project, I lead the Ecoepidemiology division. And I also lead for 1 year now a COAST action. COAST is a European Union funded grant which supports networking activities. So as you can see, there is a balance because I have the funds here available for my lab and people, and I have the networking tools that COAST is providing. Just very briefly, this pragmatic action is from the abbreviation Prevention, Anticipation, and Mitigation of Tick-borne Disease Risk, applying the DAMA protocol. And we have various working groups. I am heading the fourth one, which is citizen science involvement in the DAMA protocol. So this is where we come back to the title of the today's talk. But before we go into the details of ticks and DAMA, first of all, I want to make it clear what are my real research interests. And I could formulate it to lay people, not to you, but if I talk to children, for example, my research interest lays to find out what does the wind do when it stops blowing, or with other words, what does a pathogen do if it is not causing disease? Because I'm not a physician, as you heard, I am a disease ecologist, so I'm interested in what these guys are doing when they are not causing the disease. So this is actually a common interest between myself and Dan, because when we first met, we realized that although from different points of views, but we are doing very similar things, and we are interested in very similar things. And he introduced me to the parasite paradox phenomenon, which you all know very well, that the pathogens are ecologically said specialists and strongly co-adapted to their hosts. This is the mainstream stand of view of most scientists. But we can also observe that emerging diseases occur rapidly, and the shifts to the unrelated hosts are really common. So something is wrong with this classic co-evolutionary model, which tells us that parasites become more and more specialized to their hosts with time, and the host switches are rare, and there is a need for a genetic change in order to colonize new hosts. The problem is simple and not small at all. The problem is that this classical model is wrong. So we need a new model, and this new model is brought to us with the help of Dan, Walter, and Eric, and other colleagues as well. They introduced me also to the world of the two different kinds of emerging infectious diseases. There are the low probability, high impact EIDs, which are the most frightening, and which are the targets of most international initiatives, like the One Health projects. But these are not alone. Not only these exist because we have the so-called higher probability, low impact EIDs, and low impact is of course in brackets, because if we consider this long-term presence of these pathogens in the ecosystem, we will see that the impact is not low at all. What happens is the so-called pathogen pollution. Whenever a new host pathogen association develops, there will be first an acute infection. Usually there will be a selection for resistance, and then a chronic infection will develop, which always retains the potential for new and new acute outbreaks. So once a pathogen or a parasite, a microbe is established, it will never disappear. Some words about pathogen reservoirs, also a common interest between the DAMA people and myself. As Odi already formulated long ago, geographic range of a pathogen always exceeds the geographic range of the disease caused by it. Why? Because there is a so-called silent spread. There are always asymptomatic carriers. There are always misdiagnoses or the undiagnosed diseases, and the reservoir animals, in many cases, usually mammals and birds, which are wide-living ones, are the best pathogen reservoirs. They usually do not produce any symptoms, but they can circulate the pathogens in nature for a long time. And there it comes to the new paradigm, which will change or switch from the classical co-evolutionary paradigm. And this is based on ecological fitting, which was formulated by and used by Janssen in the 80s. To put it very simple, the traits related to the use of the broad-based resources have phenotypic flexibility and are also phylogenetically conservative. And also, very importantly, prior to the evolution of novel capabilities for host exploitation, there are always substantial opportunities for an accelerated host colonization. And here I would like to use the slides of Orsi Molnar, who very nicely presented the difference between the classical co-evolutionary model and the Stockholm paradigm. So here you can see what the classical model tells us. It tells us that the pathogens within a reservoir host need a kind of genetic change in order to colonize a new hosts. In this case, we would duly think that these new mutations and the host switches are rare, and they're also unpredictable because we cannot predict a new mutation. And as I already mentioned, this is not true because we can see it's not rare. And if it's unpredictable or not, well, we can say already that they are predictable, actually, which is, I think, the first good news during this talk tonight or today. So if we come to the Stockholm paradigm, what's the difference compared to the previous slide? You can see this very nice, colorful viruses or pathogens or parasites within the host. And this is what we really observe in nature. So there are lots of variants already existing in the natural reservoirs. And one of them or some of them are capable of colonizing new hosts. And if it's colonizing, it can be either a mammal, or even a human host, or other phylogenetically even very far away hosts as well. In the Stockholm paradigm, it gives also a very nice explanation how a pathogen which has already switched to a new host is able to colonize in the new hosts many other individuals. Just think about the COVID-19 pandemics. The explanation, to put it really simple, is slopy fitness space. This concept is based on a very simple but very important observation that the fundamental fitness space of an organism is always much broader than the so-called realized fitness space. So the first one, the fundamental, means what an organism could be doing and what are the opportunities. And the realized one is that at a given time and space, what organism are really doing. In other words, a pathogen has the same choice as humans when they encounter a room with full of apples. You either explore or exploit. What does it mean, explore? You are generalizing, reduce your fitness, reduce conflict, and in this case, fitness space will also be reduced. In the other extreme, exploitation means that you are specializing, you will maximize your fitness, also maximize conflict, and the fitness space will maximally slopy. So if we consider the Darwinian evolution of pathogens, we can differentiate the behavior of pathogens under different conditions. So within a perturbed condition, the pathogens will explore, generalize, and expand. They will increase the host range. They will expand their potential, see the ecological fitting and slopy fitness space. And the fitness space itself becomes less slopy in this case because fewer and fewer potential hosts will exist. If conditions are stable, for example, it colonized a new host successfully, and it is able to, just watching if you are still here, yes. And if they are able to exploit this new host, they will specialize, they will isolate, they will restrict their host range during time, and they will also accumulate the potential. The emergence of novel variants comes into picture, and host fitness space becomes sloppier, and there will be more potential hosts in this case. So we come to the so-called oscillation hypothesis. However complicated it sounds, it is a very simple and important phenomenon again, because if you consider the repeated episodes of climate change that we know they exist, there will be always complex mosaics of the specialized host pathogen systems, which leads to two very important conclusions: that there will be first a vast potential for repeat emergence of pathogens, and second, there will be indefinite diversifications. And this is what Dan, Eric, and Walter very importantly emphasized and structured in their book Stockholm Paradigm. They were based on ecological fitting, telling that climate change and habitat loss will always increase migration, not only of pathogens, but of their vectors and of their hosts. And this will lead to new contact opportunities and, of course, new emerging infectious diseases. Okay, so after this conceptual introduction, we all know that climate change happens right now, and it's not only the increasing temperatures, but also the extreme weather situations, whether be it floods or drought, and you don't have to go to distant places. For example, here in Central Europe, 2 years ago, there were vast storms putting cars into each other and leash, or there were vast fires all over Europe, even a hurricane in the near Czech Republic. So these extreme weather events also tell us that climate change will, of course, have a great influence onto the emergence of disease agents. In a recent Nature climate change paper, Moore and colleagues emphasized and also summarized how these different climate change events like warming, precipitation, floods, drought, and so on can influence different groups of disease agents or pathogens. As you can see, vector-borne agents are leading the group of different disease agents where climate change will accelerate both geographically and in time their occurrence. You can see that both viruses, bacteria, any kind of parasites have the same accelerating events thanks to climate change. If we add another important factor, which is also human mediated, like climate change, and this is the destruction of natural habitat or biodiversity loss, then we will also see the same effect, because humans are changing the environment, and they are giving new and new contact opportunities between natural reservoirs and pathogens and human populations. And I would like to also add another problematic factor, and this is that we only know a very tiny vicinity of microbes around us. If we consider, for example, that we know a couple of thousands of viruses of mammals and birds, and the estimations of these viruses are something like 1.7 million, then you can understand what I mean by small fraction. So there is a lot more work to do in documenting and discovering pathogens around us. And the economical side is not funnier or cheerfuler either, because before COVID, so BC, we can say, it was $1.3 trillion per year that we spent for the emergence of diseases. Usually, of course, these were the response costs for the EIDs. That is quite usual to do. And COVID itself have been already several 10 trillions of US dollars. And of course, we can very easily understand that, as in the case of individual medicine, the health organizations and agencies should in this case also do prevention instead of response options. And not only because we could save much more lives, but also from the economic point of view, it would make much more sense. Another amplifier in this EID problem is urbanization. I know that you are aware of the facts, but I have to emphasize here again that today more than half of humanity lives already in big cities, and this trend will continue. And until 2050, it will be more than 2/3 of the human population living in big cities. What's the problem with big cities? The problem is that although it seems you are in a safe haven from pathogens, cities actually are limiting factors and problematic in many ways. For example, if there is a danger, the fleeing ability of huge crowds is very limited. As you very well know, the heat island effects, which is on average 2 or 3 degrees Celsius above the surroundings, but usually or very often it's more than 10 degrees warmer compared to the natural habitats, is again a problematic part in hosting pathogens, for example. Cities, as you know, require a constant flow of energy, of water, and every kind of material goods. So that's why it is not an easy task to do all these. And if you have a high population density, of course, the kinship rates will be much lower. In other words, you will not trust trust in and cooperate with your neighbors as you would do in a smaller village. And this huge crowd leads also to an extreme division of labor and, of course, an extreme interdependency onto each other. So if you consider urban green spaces, for example, it is, again, a necessary thing. So, of course, these huge cities need these city parks, but they are also hosting many kinds of reservoir hosts, urbanized mammals and birds, even reptiles, who make use of human waste and also the green spaces that are available. And in this kind of environments, not only these urbanized reservoir hosts are present, but of course, many humans, which leads to the elevated contact rate again. You have to add only globalization, global travel and trade, and you will understand, as you are, I'm sure you do, how COVID or SARS or MERS can spread very quickly around us. And please don't consider only the North American and European big cities, but if you happen to live in or visit Southeast Asia, you can again very easily find bats and any other highly important disease reservoirs on the market. You can even buy them, whether living or dead. And in this kind of places are really the best places for switching hosts if you are a pathogen. I took part in an expert report, which was called Pandemics Report. It was organized by the Intergovernmental Panel for Biodiversity and Ecosystem Services, or IPBES. And we were summarizing all the factors that can lead to pandemics and epidemics. And for example, the wide terrestrial vertebrate trade is one of the drivers which lead to the emergence of EIDs. 24% of all the wide terrestrial vertebrates are traded globally. And with other words, we can say that we live in a minefield of evolutionary accidents waiting to happen, as Dan formulated it. And it's not just a metaphor, but if you want to show with real diseases and pathogens, you will really see that in every corner we will see a pathogen or a parasite emerging. So what is most at risk? Not the biosphere itself. It is quite clear that the biosphere, the ecosystems are capable of generating new complex biosphere even after mass extinction events, what we are also experiencing now. If someone is afraid of the species Homo sapiens, probably it is not the big danger, because we are already too numerous and different habitats to go extinct as a species. But still, it is not a good news, because the real danger is that what for example, we are doing right now, that we communicate with each other very easily at different continents with technology. This is what is at risk, because probably this will be or is already in threat with the new EIDs. So after the lots of bad news, we should find out what can we do. And we are lucky enough that we don't have to find out, actually, because we have all the resources to do the right thing to prevent the next pathogens which will emerge. And we just have to switch from the current approach to a new one. The current approach is something like the physician does if she or he has to treat a wound or encounters a disease. The main rule is to do no harm. So in case of the outbreaks or disease emergencies, let's wait until there is an outbreak and rush to deal with it, pretend there was no way to anticipate it, and then let's hope there will be never another one. But the good news is that we can be actually proactive about coping with the EIDs if we use the Stockholm Paradigm. So we have to consider these two things. If the colonization of a new host requires new opportunities, given specific pre-existing capacities, then those pre-existing capacities are predictable, which allows us to anticipate disease emergence. So you just have to use this knowledge about the different biological aspects of host pathogen interactions. With other words, we have DAMA protocol in our hands, which means we can find the pathogens before they find us. As you all know well, these 4 letters stand for documentation, assessment, monitor, and act. Again, with the slides of Orsi Molnár, I would like to show what it means. We have to know what are the microbes, what are the viruses, bacteria, parasites within the natural reservoirs around us. We have to describe them, we have to look for them. And because we cannot document everything, as I mentioned, there are probably 1.7 million viruses only in mammals and birds. And this is not possible to document decently all of them. First of all, we have to assess what we have documented. We have to find out whether they are dangerous to humans or domestic animals or crops or not. And if they are, then we have to monitor them, and we have to go back and search for the specific biological traits that allows us to act. What does action mean in the DAMA protocol? Usually it is giving advice, for example, to the stakeholders, for example, to teach citizens how they can themselves protect from disease emergence. If you're more interested, as you might already know, Eric and other colleagues have summarized recently, both in the Journal of Biodiversity and also in the latest book edition, what is the most important to know about the DAMA protocol. Very shortly, what do I mean by documentation? We have to go out to the field and we have to check. We have to check the reservoirs, we have to check the vectors and collect all the information about them. And only with the help of documentation is it possible to find the unknown microbes, which can be the next pathogens. As the WHO already called it disease X or pathogen X. As you know, if you go to the WHO website, there is a high-risk list of pathogens with COVID-19 currently on the top. But the second one is, for example, a tick-borne pathogen, clinical hemorrhagic fever. And in the end, you will see disease X. Actually, COVID-19 is already a disease X, and we have many, many others. Just we have to document and assess them. How do you document? If you are, for example, an ecologist and parasitologist in Hungary, you go out and flag for ticks. Ticks are very easy to collect because they will respond to movement. So this is what the tick flagging means. You have to move the flag on the surface of the vegetation, and the ticks will attach to it, and you can collect them, count them, and later on, of course, you can check them for pathogens, for example. You can also check the reservoir hosts. For example, lizards are very easy to catch because they don't see very good from the distance. So you can use a stick with a vinyl on it, and you can very easily catch them with this method. And lizards are very important hosts for immature ticks. As you can see here, blood-fed nymphs and larvae are all around the lateral body part, and also in the ear canal and next to the eyes. And surprisingly, they are not only hosting ticks, but also, for example, some Borrelia species. As with my former students, we identified in the 3 lizard species, there were Borrelia lusitaniae populations, which can also infect humans. So this is the way how you monitor, how you document, how you assess, and how you monitor these wide-living reservoirs. We were also studying rodents, which are very important for ticks and tick-borne pathogens. And you can also check the hedgehogs. I will talk about this soon, because hedgehogs are perfect hosts for all kinds of parasites within and also the outside. So they are a really good target for documenting what's around us. How you can assess. Assessment is actually a risk assessment. You have to check your vectors, you have to check your pathogens, and put the question whether is it a known pathogen or is it closely related to a known pathogen. If your answer is no, you can ignore for a while, but you have to archive this pathogen because later on it might be useful still. If the answer is yes to either of the questions, then you have to go back and get information about the transmission dynamics, the microhabitat preferences, natural history of this pathogen in general. What does it mean, go back? This is the next step in the DAMA protocol, to monitor. And if you want to monitor something, you really need budget, because you can explain in a grant that you want to detect something to find out whether it's dangerous or not, but it's very hard to find funding for monitoring. If you say you just want to do the same thing for many, many years. And I have the luck now that at least for 4 years, and hopefully also later on with some other projects, I will be able to monitor the different vectors and reservoirs that we started to monitor already. Monitoring means to check the eco-epidemiology of these priority pathogens that turned out during assessment that they have to be assessed and monitored. We focus on the identification of reservoir hosts because they are the key elements in the cycle. And we have to consider also that some geographical or habitat interfaces are more important than the others. For example, when people meet wildlife, this is usually the green spots in the city, or the urban wildlife interfaces are very important to highlight and to check. And of course, we can here use citizen scientists because they are very important in not only gathering data, but also this is the way how we can teach them how to prevent things. And the last step, action, is actually coping and cooperating. We have to teach citizens how to reduce the chances of establishment. We have to reduce the risk exposure, recognize the arrivals of new pathogens, and we have to mobilize every possible organizations, universities, NGOs, and so on. We have to provide proactive suggestion to decision makers. This is a very important point, because we scientists will not be able to change everything, but we are able to provide those suggestions that are necessary for change. And of course, what we scientists are really good at, cooperating with our colleagues. This is what we are doing tonight or today as well. Okay, after this still introductory part, I would like to show you DAMA in practice. How can we use it in real life? I want to show you just two examples. The first one is the urban tick-borne pathogen and the DAMA protocol. So within Budapest, I don't know how many of you have already visited Budapest, a very nice island. And this is not only an island, but also a city park. And we have studied in this city park for 3 consecutive years. What do these guys, hedgehogs, do with the parasites and pathogens? This is the northern white-breasted hedgehog, a very cute animal, which is not only looking nice, but can also bite and is very sticky. It rolls up in order to protect itself from predators. And as I already mentioned, these guys are perfect for end-to-end ectoparasites as well. So to show you how DAMA protocol went on, the first step was documentation. When we first went out with a couple of parasitologists and ecologist colleagues, we already faced the first problem. This roll-up mechanism, which is enhanced by a very strong muscle of the hedgehogs, is so strong that you cannot simply open these guys. So in the first year of the project, we had much lower tick numbers because we could not reach the most important part, the belly and the legs of the hedgehogs. So for this reason, we had to find a hedgehog anesthesiologist, which you can think it's not a very easy thing to find in every corner in Hungary. But we had the luck to work with the single person who was able to do it, I think, in the country. He was that time the chief vet in the Budapest Zoo, Viktor Molnár, and together with many colleagues and citizen scientists and people interested, we were setting up a small field lab, I would call it, where we had not only the headlights, but also some lights. We had a table, and we could anesthetize the hedgehogs. And if you anesthetize a hedgehog, then you have safe haven for collecting parasites, because in this case, you just need actually patience in order to remove many ticks. We of course marked the hedgehogs. And with this, we could also check what happened with them later. It's a night shift, so in order to collect hedgehogs, which are nightly active, we had to go there after sunset. And after sunset, we could grab them by hand and take to this center where we could remove the pathogens. We also collected blood and tissue samples from these guys. As you can see, sometimes these guys are among the quills, but more often, this is actually the exception, but it looks really interesting. The most of the ticks, as I mentioned, are on the belly and on the legs. That time we didn't call them citizen scientists, but they were at some nights we were 40 to 50 people, very enthusiastic students and friends and colleagues who were collecting the hedgehogs. And we, the parasitologists, could collect all the samples. During the 3 years project, we collected nearly 10,000 ticks, and on average, it was more than 30 ticks per hedgehog, but on some of the hedgehogs, we found over 200 ticks. These were larvae, nymphs, and adult ticks as well. And most of the hedgehogs were infested with at least one tick. We published the results that time, but we went on and we wanted to actually assess the threat, what these many ticks mean. And our first important observation was that although the natural cycle of the tick, this is the sheep tick, I forgot to mention, Ixodes ricinus, involves wild rodents and wild living birds and middle-sized mammals and larger-sized mammals. 3 hosts are needed for the 3 stages. But in this case, we found out that because there are no larger mammals on the island, hedgehog itself is enough for all 3 life stages. So this is the perfect natural host in this case within the city park. Among the 10,000 ticks, we also found a single Hyaloma marginatum nymph from a hedgehog, which is very interesting because it's not endemic in Hungary. And later on, I will talk a bit more about this species. So after collecting this huge amount of samples, of course, we had to identify the ticks, we had to extract DNA and do molecular detection of pathogens. We did it with a collaboration with Dutch colleagues, actually. And we had really interesting results. For example, the Lyme disease agent, Borrelia burgdorferi sanzolato, in Europe we have different genospecies. Actually, nearly 100% infection prevalence was observed. So we had positive hedgehogs and only positive hedgehogs with some 1 or 2 exceptions. Another pathogen, Anaplasma phagocytophilum, is also very prevalent in the ticks, and many others were also checked. We published also some of the results already in Emerging Infectious Diseases. And later on, we put the question, okay, we have these many pathogens and many ticks in the hedgehogs and on the island. But what about the real risk of infection? As you know, there is a very simple equation. Risk equals to the hazard times the exposure with the pathogens. What does it mean? Hazard is actually a chance of being infected. By the way, we had to screen the flagged ticks for pathogens, and we've done monthly flagging of ticks for a whole year. And we also tried to examine whether different patches on the island have different prevalence of pathogens. And as you know, exposure means what are the chances to encounter infected ticks. And in order to find this out, we made an online questionnaire which targeted the frequent visitors of the island, like joggers and the hedgehog collectors themselves. What it turned out is that it's not only the hedgehog, but also the ticks in the island. So those ticks that are questing, looking for hosts and are able to bite humans are also very much infected. Around 1/3 of them was infected with Borrelia. A very high percentage of them were also infected with Anaplasma phagocytophilum, and also Neurechia mycorrhizis. The questing ticks were found only in those areas where there is a dense vegetation and not on the mown grass. And the questionnaire showed us that although some of the island visitors know that there are ticks on the island, but most of them did not meet ticks at all during their visit, and nearly 2/3 of them told that there are no or very few ticks on the island. So what does it mean? It means that there is a huge hazard because there are lots of infected ticks on the island, some of them carrying many pathogens, but the exposure itself is low. So this is the reason why not many Lyme disease cases occur, for example, in the local GPs, because the people do not encounter the ticks. And we had a hypothesis that actually there is a marked habitat difference on the island. And we knew it very well because whenever we were flagging the ticks on this short mown grass, we never found or hardly ever found ticks. You could see here the numbers. It was 5 ticks altogether during 1 year. However, we found many more in the bushy areas. What does it mean? It means that you have to encounter the ticks if you go to the bush. When do you go into the dense vegetation? If you go when you have to go. And this is what we found out, that the biggest risk is actually in a city park when there are no public toilets. And here we come to the action part, because there is a very simple recommendation for the park maintainers. Please settle and provide public toilets to a city park. And in this case, you can easily reduce Lyme borreliosis, anaplasmosis, whatever kind of tick-borne pathogens. And at that time, when we did this study, there were only, I think, 2 functioning public toilets. And I tried to put this data and tell this at every possible opportunity. And now I can tell that there are around 6 or 7 public toilets already, and I hope it will be more. So this is how you do the action part of DAMA protocol. And of course, the story doesn't end here, because you have to start over and over if you deal with DAMA protocol. For example, we were interested if hedgehogs in general are also so much infected and infested at other places. And because hedgehogs are protected animals, we found out a very easy way how can we get access to tissues of hedgehogs, and these are the road-hit mammals. So we started another project where we were interested in any kind of possible reservoir hosts, like not only hedgehogs but squirrels, which are also prevalent in city parks, of course, considering written consent from every single squirrel. And by collecting the road-hit rodents and also hedgehogs, we very easily got access to, again, many pathogens and parasites. Here on the right lower corner, you can see the ticks removed from a single hedgehog, road-hit hedgehog. And here, this very ugly looking thing, which also went later on to the cover of a journal, is actually what we call a hedgehog sticker, because it's actually two-dimensional. Very often within the city center, buses drive through these poor animals. And we were still interested whether it's possible or not to extract DNA and to do PCR of pathogens. And these very dry, old, bad condition carcasses were also a perfect object for pathogen detection. So it's a very useful method actually to do the documentation and the monitoring phase. We also designed an online map, and it was possible to put what kind of species of mammals were collected, and also to show what kind of pathogens. As I said, although ugly, it made to the cover of the Transboundary and Emerging Diseases journal. Okay, as I promised, there is a second example how we can do DAMA in practice, and this will show us how citizen science can produce very valuable data to do the DAMA protocol. To put it simple, we have to use the enthusiasm of people that they have in scientific projects, because if you look around, people are really interested to do something. They give the hand. You just have to say that I need this hand and I need this help to my project. So we wanted to make use of this citizen science method in finding out whether hyaloma ticks are present in Hungary or not. Why is Hyaloma marginatum, for example, important? It is important because it can carry very dangerous pathogens, I will show you later on, which are of public health concern. But first of all, I want to show you the life cycle of this tick to understand how is it carried to Hungary and why it is possible to encounter them in Central Europe, where it is, or it was before, not endemic. It is a two-host tick. It means that when it hatches from the egg, the larva is looking for the first host. It is usually a small mammal or a bird, and if it's a bird, this is the problem, or this is the way how they can do big distances, because when it is fully fed on the bird, it will not drop off like most ticks, but it stays on the bird and it molts to the nymphal stage. And this nymphal stage, the fully fed nymphal stage, will drop off the host. But this takes several weeks to reach. So this means that they can fly from Africa or the Mediterranean to Central and even to Northern Europe and drop off there. And if it drops off, it can molt to adults. And here it comes to climate change, if climatic conditions allow, these adults will develop and will be able to find, for example, ungulates or humans to feed on, and even they can start a local population. So these migratory birds are key in this cycle because they are able to carry the immatures of Hyaloma ticks to the northern parts of Europe. And why they are important? They are important to check and to monitor because they can carry, these ticks can carry Crimea-Congo hemorrhagic fever virus. So the name is already frightening, but the disease is even more frightening. According to physicians who have seen already patients suffering from disease say that it is even uglier than Ebola, actually. The rate of death, in case of CCHF, is around 30%, so a bit better than Ebola, but it can lead to hemorrhages, it can lead to inner and outer blood developments. So this is a really frightening disease, and there is no cure, no vaccine against CCHF virus. So this is why we monitor the presence and spread of hyaloma ticks. And in the recent decade, many observations were made, for example, in Germany in this case, but all over Sweden as well, so very north, that not only the immature ticks were carried to these parts, which we already knew for 100 years, but also the adults were observed. And this is really frightening trend, because the adults sometimes bite humans and they are able to carry the virus itself. So this is the reason why we started the Citizen Science project. This is a Hungarian website originally, which we started in 2021. And later on, we went a bit more professional with the help of a company. And we already have this English website as well. And with the help of the website, we were trying to persuade people to look for these ticks. We are lucky, actually, because hyaloma ticks are something different. So here in the middle, you can see what 99% of humans know as a tick in Hungary, before feeding at least. This is the sheep tick, Oryxodus ricinus. And if you have a dog or a cow or a horse, you will also observe sometimes these dermacenter ticks with the patchy scutum. But this guy is even bigger than the dermacenter. It is 2 or 3 times bigger than this common tick, and it has stripes on the leg. So it is not only big, but it is also very easily differentiable from the other species. And I want to show a video that it is not only big, but it is also running very quickly. This is how it goes. So in contrast to the local tick species, which are very slowly crawling up to your leg and you don't see it, these guys are visible, and these are really fast. And they are also able to perceive the carbon dioxide that you exhale for several meters, and this enables them to find the host very easily. And this is why during a citizen science project, we were lucky enough to collect these ticks. First, we asked people to send in photos if they see such a tick, but we also asked them to send the specimens to us, because we were interested not only in the hyaloma actually, but because people were sending samples from all around Hungary. With the help of this method, we were able to cover actually the whole country in the sample collection, which otherwise we weren't able to do at all. Okay, so back to the project. We were telling what people have to look for, and of course we were also telling them what preventive measures they have to do. And of course, we didn't want to frighten people, but the CCHF, the virus itself, helped us a lot. I think that people really were curious about the occurrence of these ticks. Of course, we monitored with the help of Google Analytics the website users, and it was a real success, I can tell. People started to send in different kinds of arthropods. Of course, different insects and beetles and spiders and everything. But after a while, of course, we also received some decent ticks, like here, the most common sheep tick, and here to the right, the dermacenter tick, probably from dogs. We also made or used every possibility to spread the word. So we were trying to tell people that we are collecting these ticks. We went to any public outreach opportunities. Here's Eva Sabo, my grad student, showing the lay people what they have to know about ticks. And we made many, many interviews with national television, radio, and online news outlets, because this is the way how you spread the word. And you have to be careful with the media, because sometimes they use the wrong picture. For example, this one is not hyaloma, this is the DAMA Center, and because this is a website that is visited by many people, many people sent after this publication of the photo many DAMA centers to us, like this one. Of course, if you collect samples, you will see strange things. So you have to consider, for example, the GDPR, how we call it in the European Union, so that the personal data never go public. For example, in an online lecture, in the mentor seminars, that someone has a size of 16. But of course, it's not the most important. The most important is this tick on the other side. I also received soft ticks, which is not so easy to find. So it is a really good project, because the side catch is also important and interesting. Some mothers from schools sent very nice photos from the children's eyelid, where a tick can also occur. And these are all of them very valuable data. But after a while, we also encountered the striped-legged hyaloma ticks. So already in 2021, we were able to find, with the help of this enthusiastic citizen scientist, hyaloma specimens. The first couple of them were from domestic animals, and later on it turned out that all of them usually comes from domesticated animals. This one is a hyaloma rufipes from cattle. It can also spread Crimean Congo hemorrhagic fever virus. We were able to do it with collaboration of Gabor, the virologist colleague. We were not only looking for the Crimean Congo hemorrhagic fever virus, but we're also interested in the phylogeny of these specimens. So we were checking mitochondrial genes, and we could establish that this belonged to the European African clade of Hyaloma rufipes. And another one from a dog observed from a dog was Hyaloma marginatum. This is the most important vector species. And we also have done the phylogenetic analysis, and this belonged to the Eurasian clade of these ticks. We received also samples from donkeys and horses. And as I mentioned, the side catch was also interesting. We published the results of the first year of this project in transboundary and emerging diseases. And you can see that we received really from all over the country. The first two hyalomas you can see here to the western side of the country and here in the south. We checked all the specimens for CCHF. We have found no CCHF, but we have done also a deep metagenomic analysis, and we found something else. Here you can see a huge tree of the so-called Bunya viruses. Bunya viruses are the big order of viruses where Crimean Congo hemorrhagic fever virus also belongs, here somewhere. And we found a tick virus, which has only found in a single case here somewhere in Russia, and it has been not described. There were just 2 hits in the gene bank. So based on the Russian name, we have to call it Voltskoe tick virus. And we characterized it for the first time with the help of virologist colleagues. And this led to the publication, which has been submitted 5 months ago to the Emerging Infectious Disease journal. You can guess what happened. Nothing so far, but at least we are still in the place, so we are waiting for reviewers. Okay, and here you can see the hyaloma results so far. You can see that it is not only single locations, but also there are some aggregated places. For example, here at a buffalo farm, there are already 5 hyaloma sightings. So this is very important, because it is the first hint that probably it is not only introduced or emerged here, but there is a very high chance that it is already established there, and there will be, or there is already a local population of Hyaloma marginatum. Okay, with the numbers, I just received today the third one this year. So we have 3 hyaloma sightings and specimens this year, and altogether we have already 12, and many others from other species. Okay, very shortly, we have a new project which also includes citizen scientists, Tick in the Garden project. In this, we are asking people living in cities and towns to collect ticks on their own. Because we are a small research group, we cannot go to many city gardens, and we are interested how people can infect it within their own gardens. So we produced a small video showing how people have to use their flag, which we are sending to them, and how they can collect safely the ticks with the help of these tapes, and they can send in the samples, something like this, with the date, the weather data, and how long they were flagging. And then we can use these specimens for not only tick occurrence, but also for pathogen prevalence studies. It's a huge work, mainly done by my grad student, Eva. We sent out already nearly 500 packages with the flags, and people will flag for us on 600,000 square meters in more than 250 different towns and cities, which is really very interesting. And we hope that that next time I can already tell what kind of results come in. Okay, so to close the circle, we have to go back again. So we have to document, act, monitor. We have to document, assess, monitor, and act all the time. And of course, it is not a stable project. So as Dan and colleagues formulated in the book, the protocols themselves have to be evolvable, because there is no static solution for the problems that involve evolving Earth and evolving biosphere. So this is what we try to keep in mind with our resources. And our aim is to do an operational One Health, which is badly needed, which includes not only the pathogens and the vectors and humans, but also the natural environment, the natural hosts, and the domesticated animals, and not to forget the crops and the crop diseases as well. And if we reduce anthropogenic global environment, then we can environmental change, then we can reduce pandemic risk. And also with the EBES study, we concluded that land use change, agricultural expansion, and urbanization can cause more than 30% of disease emergence. So what we have to do is a so-called transformative change. So we have to find the critical knowledge gaps. We have to document much more microbes and pathogens than we do now. It would be very important to establish an intergovernmental panel also for the pandemic preparedness involving the DAMA protocol, as the IPCC or IBES already exists. It would be very important to introduce economic incentives also for natural protection and reducing pandemic threat. We have to stop habitat destruction right now, and we have to reduce wildlife trade. And very importantly, we have to use a very broad societal involvement. And this is why citizen science is very important, because it not only gets the data to our lab, but it can also teach and educate the interested people. How can they reduce the disease emergencies themselves? So in other words, our only chance is to change. And with these words, I would like to stop my talk. Thank you very much for your attention.
Wow, amazing presentation, Gabor. Thank you so much.
Yeah,
Wow.
thank you. That was absolutely spectacular. In fact, that's a major lesson for people on how to make these presentations.
Yes.
Thank
It
you.
really needs to be distributed as widely as possible. You know, that's ideal material that could easily go up on YouTube and used in classes around the world. So no, that was spectacular. Thanks.
Thank you.
Thanks, John. Any other questions or comments? We have a few comments on the chat. So can you see the chat there, Gabor? I'll see if I can get at it.
I'm just having a look at it. Yes.
Yes. That was amazing. Really good and well explained and exciting.
Yes, thanks. Yes, I think the second question, yes, it's very important. And I very often get this question, how do I make sure that the citizens are not putting themselves in a high risk? So we try to reduce this, for example, by telling them that, for example, in this Tick in the Garden project, the ticks are there anyway. So people are going to their gardens and they are spending sometimes many hours per week there, collecting the leaf litter, doing whatever garden work there. And with the help of this citizen science project, we are actually calling attention to this danger. So I think we are not putting them to danger, but on contrast, we are saving them for going to the disease risk area, which is their own garden, and not preparing that they can be infested by ticks. So if we are collecting, or someone is collecting in his or her own garden, I think the best we can do is to tell them that there is a risk. And if they are do it with a flag, for example, then they have a chance to find these ticks and collect for us. And we always emphasize that they have to check themselves for the ticks. The repellents. Yes, very often I got the questions, but the problem with the repellents is that it's never 100% protection. So that's why I usually tell the people, yes, you can use the repellents, but you still have to check yourself for ticks, because this is nearly 100% efficacy. If you remove the ticks on the same day, the pathogens have no chance to develop the disease.
Very good answers and a good explanation. I think that alerting people to the prevalence and to the presence of these ticks in their yards is really important. We have a lot of them in our backyard, even though there's very few mammals coming through. We have squirrels and rabbits, and that's about it. But there are ticks there, and I've discovered several of them there.
Yes,
So
squirrels and rabbits are perfect hosts.
yes, yeah, it's a good one. So let's see. There should be another one here.
Yeah, are there sightings after the start of your study near large cities? Does something for college with people? Of course, we haven't started receiving many samples, so I cannot tell any results of the analysis. But definitely it depends a lot, not only on density, but in case of citizen science projects, very often it is biased by education, even biased by income of the people, because the more wealthy and more educated people are, the more they will tend to take part in this kind of citizen science actions. This is actually an obstacle, because we do not reach those people, for example, in the villages who are having farms and having cattle and horses around. So we need to use different methods to reach different people.
Very good. I think that a broad spreading of ideas is important. Being on the radio and being on television makes a big difference. Here's another question from Sasha. She said, how would you target the rural human population? So how do we actually get to people out in the farms and in the woods? Over.
It is a very tricky point, and as I just mentioned, we encounter this problem. It's very easy to reach people who are using the internet, who are watching the news, and whom we can reach with the interviews. And you have to use a completely different mechanism or strategy if you want to reach people who are not using the internet. In case of, for example, cattle producers, currently we think the best way to do is to print an A4 page and just put to the stables and make a big picture of the hyaloma tick, for example, and show that we are looking for the striped-legged big ticks, and put the phone number, for example, there that they can call us, or maybe an email address. This is possible, and this is working actually. This is what I've already tried.
Very good.
In some cases, for example, simple Facebook groups are possible way for reaching these people, because although they are farmers, they are not spending a lot of time on the internet, but they have their own private groups on Facebook. And if you have access to it, because they have some problems with ticks, then you can call their attention about this project there.
Very good. I think that's an important part. I think going to county fairs, if they have state fairs, county fairs, those would be good to distribute there too.
Yes, yes, that's right. That's right.
Okay, another question that came into my head was, I saw that you had the picture of the world with emergences, and it looks like North America has more emerging diseases than any place else. Is that because we have more people flying in from all over the place, or is it because we have more money to look for these things, or a combination?
Both, it is the usual research bias or research intensity. I think both. So it is density of people, also the grants and the money which is available for this kind of research. I think both. But the real disease hotspots so far, at least for the low probability, high impact EIDs, is still Southeast Asia and tropical areas. And of course, in these places, the knowledge is the lowest, actually. So there are huge knowledge gaps over there.
So it's happening, but we don't know it.
Sorry?
It's happening there, but we don't know it yet.
Yes, yes, that's the case.
Yes, right.
I have a
Question.
so pedaling off of that, when looking at that map, it looked like the southeastern corner of the United States had the most density of those. Is that possibly because there's a reflective climate between Southeast Asia and the southeastern part of the United States, and how maybe those two would be similar environments for availability for spreading or exposure, maybe?
The problem in answering this question is that it's complicated. As you know, there are so many factors, because you don't only need, for example, high population density of humans, but you have to also you also need a situation where the pathogens or potential pathogens and humans can encounter. And you need also these wide living reservoirs. You need, in case of vector-borne diseases, the vectors that are able to take the pathogen from one host to another. So it is always a unique combination of these factors where something emerges. And this is the reason why we have to do the DAMA protocol, because without understanding the local and very specific interactions between these natural reservoirs and pathogens, the pathogens and the humans, we have no chance to forecast or prevent anything.
Exactly. That's a pretty good explanation. So I have one more question. And the question is, are viruses exclusive in a tick? So if you have one tick that has a certain kind of virus, is it decrease or increase the probability of that tick getting a different type of virus or a different type of rickettsia or other spirochete type of bacterium? Is it something that we can know?
Yes, there is an influence, but not in a direct way, because the tick is a slow feeder. So it is feeding for a single host in a single stage, but for many days. For example, a female tick is feeding for up to 10 or 14 days, depending on the species. For this reason, if there is a virus in a tick specimen, it always depends on the previous host, what kind of other pathogens or microbes it gets. So for this reason, some pathogens are connected with some other pathogens simply because they come from the same reservoir hosts.
Yes.
I don't know if the question was this.
Oh, that's a good explanation, and I think
Okay.
it's a way to think about it.
On the other hand, of course, microbes influence each other, but that's a completely other story. It's a very interesting story, because ticks have their own microbiota, and some microbes are needed, for example, for the digestion of blood. And this is again a way actually how we can fight ticks, because some vaccines already target this microbiome. It's a very interesting topic as well.
Right. It was kind of a bipartite question. So you answered both of them. Thanks.
Okay.
Okay. Anything else here? I don't see anything else coming in. So, Valeria. So Valeria, go ahead. I see you waving your hand. Go ahead. Sorry.
Sorry. Thank you, Gabor, for this presentation. It was really nice. I just been thinking lately to a lot for my study model is a vector-borne disease, vector-borne pathogen as well, about host manipulation by pathogens. So I want to know if you are aware about any example where pathogen induce any changes in ticks that may affect pathogen spreading, distribution, and so on, or host association as well.
Yes, a very, very good question. Thank you. And, you know, it's such an amazing topic. Of course, there are examples also for this. I think Russian scientists observed for the first time that tick-borne encephalitis virus, which is very important virus in Eurasia, is able to change the behavior of the infected ticks. In a very simple experiment, it was shown that the infected ticks with this virus were quicker in the experimental setup. With other words, those ticks that carried the virus had higher speed and were finding the host quicker. So it's really interesting because it acts synergistically actually with the tick, and in a way they collaborate, we can also say, and the infection itself helps the tick find the host.
Wow, that's amazing.
It is really amazing. And there is an even more amazing one. In this case, it's actually the US Lyme disease agent, Borrelia burgdorferi. One of the most mind-boggling example, I think, in case of tick and pathogen interaction is that Borrelia, you know, which is a bacterium, a very simple and primitive prokaryote, is able to interfere with the gene expression of the tick itself, and it can change the gene expression of the tick and provide or make the tick produce a protein like a coat which will cover the Borrelia, and with the help of this coat it can evade the immune system when entering the rodent. So this is something really amazing, I think, that the prokaryote will let the tick produce something in order to protect itself from the immune system. So again, they collaborate because both the tick reduces or manipulates the mammalian immune system, and the bacterium is making use of it.
Great. Yeah.
That's amazing
Thank
too.
you.
So, wow. So your phytoplasmas do that in their leaf hopper?
Yes, they do. They do. And I've been thinking, how do you consider this? Because I read someone saying that because the pathogen is not causing any harm to the vector is not a host, is actually a carrier. But I see this differently, because in the example that you just provided is not just commensal association, it may be also mutualistic association or something like that. So I would consider that definitively an host. So I don't know what you think about that, but there are more conversation around this kind of association and consequences for pathogen to spread across the environment.
Yeah, we could have a whole discussion seminar on semantics on this one. That would be there we go for a parasitology course. So
yeah.
All right.
Yeah.
Well, thank you.
Thank you.
Anything else, Valeria?
No.
All right. Well, thanks a lot. We appreciate it. And so Gabor, thanks for being with us today. Anybody has something else, raise a hand. Otherwise, we'll let everyone go. And Gabor, I went ahead and recorded this. And if you don't mind, I'll go ahead and post it to our Facebook page.
Of course, I'm happy if you can do that. Sure.
All right. Well, thank you so much. And I hope to meet you soon in person.
Yeah, me too. And it was my pleasure. And thanks for watching and hearing, listening.
Yes, it was amazing. So thank you so much. See everybody later. See everybody next week.
Bye.
Bye-bye.
Bye-bye.